So today's lesson is about Punnett squares, which for many of you, at least the simple ones, will be a review. Some of you may never have done them, which is fine. They're pretty simple. We are going to look at more complicated ones, though. Right? We're also going to talk about the purpose of the Punnett square, because a lot of times people don't realize why we would use it. Right? It's not just fun or some extra thing. It actually has a purpose for predicting. Although I personally think they are kind of fun too. So we will define examples of dominant and recessive traits in humans. And we're going to use Punnett squares to predict offspring using one or two traits, which is what we're going to talk about. So hopefully you've gotten a chance to go through the Mendel lesson from yesterday. If you didn't, that's okay. We're going to quickly review some of the content there so that you can understand what's happening right now. Otherwise, go back to that lesson. It has a lot of vocab. The hardest part about genetics is understanding the words, because they're going to use a ton of words to explain. And if you don't know what those words mean, good luck to you. So some helpful vocab. These are the ones that we will mostly be using today. Again, these might be words that you are familiar with. You probably touched on this in middle school somewhat, right? Depending on your teacher, probably. But we have our dominant traits and our recessive traits, right? And we know Mendel is actually the guy that figured this out. I don't usually do a lot of learn about the scientist stuff, because then it just becomes depressing because they're all white men. And you're like, oh, well, that's what science is, or was, but not anymore. But Mendel is actually super fascinating to me for two reasons. One, he didn't know what DNA was, and yet he found out all this stuff about DNA. Super interesting. And two, he literally just had a book where he wrote down observations about plants, and then he happened to notice that there was something going on. Right? How many of you guys have a journal where you document what your plants are doing every day? Probably not that many. Although Lane might, if Lane's here. But still, right? A very interesting guy. So he came up with this idea of the dominant and recessive traits. And dominant means that it is expressed no matter what. If you have an allele that's dominant, it's expressed. If you have an allele that is recessive, it will only be expressed if you have two of them. And we'll review the word allele as well. We represent the genotype, at least, of these with letters, right? And the genotype describing what the genes are. So if you see a capital letter, it means it's a dominant trait. If you see a lowercase letter, it means it's a recessive trait. Now, I could give you letters and not even tell you what they are, and you could still figure out what would happen to the offspring, right? But it's helpful to know what the trait is representing. From the lesson, T's, right, were for tall and short for the plants, tall being dominant, which is why we use T's. Now, both of the letters have to be the same. You don't do a big T for tall and a little s for short. Right? There are some situations, you've been crossing beans for years with no written diary. See, you're missing out. You could be Mendel part 2. So you have to use the same letter, because that tells us that we're talking about the same gene. Now, there are some genetic situations that we're really not going to touch on this year because they're not required. But there are certain situations where traits can both be expressed, where they're co-dominant, right? So both of them show up. There's also situations where they don't necessarily show up, but they create a new thing together when they're both expressed. So there are other situations. Okay. Oh, good. We're glad you're here, Lane. All right. So then my question to you guys is this, which if you look through the lesson, you should know the answer. Is every gene like this? Does every gene work this way, where it's dominant or recessive, and it's expressed or it's not? No, right? I just gave a couple of examples. One that's pretty easy for you to think about is the color pink in flowers. Pink flowers are usually because you have two dominant traits, red and white, both being expressed, and together they make pink, right? Whereas if you had two pink alleles, it would be pink. And if you had two white alleles, it would be white. If you have a pink or a red and a white one, you get pink. Sorry. Red versus white. Gotta get my brain around this. Another example would be sickle cell anemia, right? Is a great example where you can be heterozygous. You can have some of your cells that are normal shaped and some that aren't. And that actually is beneficial for you if you live where there's malaria, because you won't get malaria, right? So maybe you can't breathe oxygen as efficiently as somebody with 2 sets of alleles that produce regular red blood cells, but you won't die of malaria, so it might be something that's good to have in certain areas, right? So that's kind of how genetics works. So yeah, hair, okay, so hair type is one of those ones. So there's a lot of, this is the thing too, there's a lot of fake news out there about genetics. A lot of fake news. In fact, a lot of the things you've heard about genetics are probably wrong. Hair is not that simple. Hair is controlled by many genes. There is one gene that potentially could control straightness or waviness. However, we know from looking at people that there are not 3 textures of hair. There's like 1,000, right? Same with eye color, right? There's not 3 eye colors. There's a lot. Some people's eyes are like super blue, and some of them are kind of gray blue, and sometimes they're greenish, and sometimes they're brownish, and sometimes they're this, right? The reason why is because there's many genes contributing to that. So a lot of traits like height. Sometimes people say, well, both of my parents are really tall and I'm so short. Yeah, right? Because height is not inherited this way. It's not a dominant recessive situation. It's controlled by many genes. It's going to depend on which combination of genes you got. So many traits are polygenic, right? Meaning many genes, polygenic. Control. So it can be really hard when you start breaking this stuff down, right? Like sometimes people are like, oh, I must be adopted. This didn't work, da, da, da. No, it's probably not a simple inheritance pattern. Pea plants are pretty simple. Most of their traits, they have 2 choices. It's this or it's this, right? Humans, it's not quite as simple as that. Okay, so I will say that a lot of the stuff you hear is fake news, so be on the alert. Research, right? Find at least 3 sources that say the same thing, that have real science backing it up, and then you can go, okay, I think this is true. Yeah, so eyes, hair, height, right? Those are great examples of things that don't work in this way. So what things do work in this way? Because of course we always want to relate this back to ourselves. Trait examples that do work this way. Widow's peak, right? That's actually the presence or absence of a peak of hair. So I don't have one, right? My hairline is pretty straight. Some people have a super exaggerated one, right? Like a Dracula widow's peak, or it could be just subtle. But having a widow's peak is a dominant trait. If you have a straight hairline, it's recessive. Right? Interlaced fingers. Put your hands together. Look at your thumbs. Which thumb do you put on top? Right? I put my right thumb on top when I interlace my hands naturally. Having your right thumb on top is recessive. Having your left thumb on top is dominant. I have a lot of recessive traits, actually. Dwarfism. Right? Maybe you know someone who's a dwarf, or you've seen a dwarf before, right? Dwarfism is actually a dominant trait. Again, going to show that dominance does not necessarily mean that it's more common, right? Do we see 75% of our population being dwarfs? No, right? In fact, we can talk about that if you guys want. I'll give you an example of why that works, because if you have 2 dominant alleles for dwarfism, you actually will die, right? So homozygous dominant people can't exist there. It results in death. That happens a lot with genetics. Certain combinations, fatal. Hemophilia, you might have heard of it, right? Russian czars, big problem. You get a little bruise and you bleed to death. Kind of a big deal, especially without modern medicine to help you, right? That is a recessive trait. And it's X-linked. What does that mean? Again, it's something we're not necessarily going to go into in this class just because we're running out of time, and I'm so sad. Because I would have spent probably like 3 months on genetics if allowed. But X-linked means that it travels on the X chromosome. So X-linked means that if you have XY as your chromosomes, right, male chromosomes, if you get one X chromosome that has this recessive allele on it, you get hemophilia, which is why it's more common in males, right? For a female to get it, you would have to get 2 X chromosomes, both with the recessive allele, and then you would end up getting hemophilia. Same with color blindness, right? Same with baldness. Also, baldness comes from the mom's side of the family. So if you're a dude and you're worried if you're going to go bald, look on your mom's side. If there's a lot of bald people, you should be afraid. In fact, I used to make it an assignment, like a Thanksgiving assignment, when I used to teach genetics around Thanksgiving time, to like go to Thanksgiving and try to predict who was going to become bald. You know, as like a little extra credit, because some people were like, oh, that would be hilarious to just go up to my uncle or cousin and be like, yeah, you're probably going to go bald. Here's why. Okay, so anyways, continuing on. Oh, the final one is PTC, which is actually a really interesting one. This is a random chemical that someone discovered in a chemistry lab that's pretty, like, it's harmless and it's not really used for anything, but the ability to taste it is genetic, right? I actually have some PTC papers, so if you're ever at the school and you want to try it, let me know. If you can taste it, apparently it tastes awful, like so disgusting that you want to throw up immediately. And if you can't taste it, like me, it just tastes like paper. So it's pretty funny because you could be next to someone and you go, oh, this just is like paper, right? And then somebody else is like, because it's the grossest thing they've ever tasted. It's super bitter. So again, a random trait that happens to be inherited in these perfect dominant recessive patterns. Yeah, so if you're around and you see me, tell me you want to taste the PTC paper and I will let you have some. Okay, so Punnett squares. A way to predict offspring. All right, so a lot of genetics questions that I like to write and that you'll see involve using the vocab so that you can get used to it. So a homozygous recessive parent is crossed with a heterozygous parent. How many of the offspring will be tall? Oh yeah, so I meant to say before this, we're going to use the backstory that I created for the pea plants, right? That we have T being tall and T being short as our traits. So type a 1 in the chat if you have done one of these Punnett squares before with 4 boxes and you're like, cool, this is going to be so easy. Okay, Marissa's done them, Caden's done them. Yeah, right? Pretty common thing. You had to teach it. Yeah, right? So again, probably not the hardest thing in the world, but the one thing that I do want you to understand is what we're actually writing on the Punnett square. What do these letters represent? Okay, first of all, they tell you about dominance. They tell you the capital letter is a dominant trait, the lowercase letter is not. Cool. What are they actually representing? Are they representing people? Yes, Marissa nailed it. Alleles. They represent alleles, right? That's how you say it, by the way, allele. So an allele is a copy of a gene. There can be many varieties of alleles, right? In our situation here, we have 2 alleles, tall or short, right? Other genes may have many choices. Again, we're not going to do those, thankfully, because it's way complicated. So these are representing alleles. So now let's break down what the words mean. What does homozygous mean in terms of a descriptor of someone? Both the same. Exactly. Right? You might even think of like a homogenous mixture or homosexual, liking someone of the same sex, right? It means the same. So in this case, we have someone who's recessive and homozygous. We're going to have 2 little t's is what that means. So that's our parent 1. 2 little t's. And again, why would parent 1 have 2 alleles? One from mom, 1 from dad, right? Even if you were made in a test tube, you're still going to have 2 alleles for every gene. Now, there are some insane genetic situations where there are 3 parents. If you're interested in weird stuff like that, or one parent, or weird things happening, look no further than the banana plant, right? Bananas do some weird genetic stuff. So if you're interested in that, research bananas. Otherwise, in all of our examples, each parent will have 2 alleles. So we have a heterozygous parent as well. We know heterozygous means 2 different alleles, right? It's obviously got to be different than the same, must be different. So we're going to have big T, little t. Now, you can write this little t, big T, right? It's the same thing. However, it's usually the culture of genetics that you write the dominant trait first, just so you'll be on the same page as everyone else in the world. So now to set up our Punnett square, we want to know, well, how many of the offspring will be tall? Well, we could look at the parents and sort of write out all the possibilities that could happen here. But what we know is that in meiosis, we create 4 gametes. A parent who has 2 little t's will create 4 sperm or egg cells that will just have 1 allele in them, right? Because remember, the parent's only contributing half of the information. There's 2 choices, but they're contributing half. Our other parent, though, their egg or sperm will be different. They'll have 50% with big T's and 50% with little t's, right? In this case, 50% with the gene for tall, 50% with the gene for short. So really, this heterozygous parent is the one who will determine what the offspring look like. So to set up the Punnett square, all we do is write the letters across the top, which most of us probably know, but now you know they represent alleles. They're not just letters, right? So parent 1, I'm going to write across the top 2 little t's. I'm going to make my t's have these little curves just so that you can tell the difference, because my writing with the mouse handwriting is kind of bad. Parent 2, we know they have 1 big T and 1 little t. So all you do is write the possibilities of alleles, right? Parent 1 can only give little t's. Parent 2, though, can give either or. Then what do? We combine them into our boxes. So we bring our big T into this box. We bring our little t into this box. There's 1 potential child. This box is going to have 2 little t's. Another possible child. This box is going to have 1 big T, 1 little t. And finally, our 4th option here will be little t and little t. Okay, so now that part's the easiest part, honestly, filling in the box. You just look at what rows and columns do you have. But now we determine what this means. So back to our question, how many of the offspring will be tall? Well, look at your offspring here. What percentage of the offspring will be tall? 50%. Why? Because 50% of them have a big T, which represents tallness. Now, tall is the phenotype, right, or what you physically see. If I asked, for example, how many of the offspring will be homozygous recessive, just like parent 1, right, the answer would still be 50%, because 50% is the only answer here, because there's just 50% of each. But 50% of the offspring will be homozygous recessive. They'll have 2 recessive traits. Now, where genetics gets interesting is then usually, right, in non-human species like plants and dogs and whatever, you could cross the brother and sister, right? So what would happen if we crossed the brother and the sister? Well, the same thing. Or we would get all short plants. So that's where you can end up having multiple generations where you're inbreeding whatever species you're looking at here. There's also a ton of interesting research on that, right? How inbreeding limits the genetic pool and causes a lot of weird stuff. So that's the basic Punnett square. Really not too complicated, but make sure you're understanding what these things are representing, right? These are alleles. These are the potential combinations of alleles that could happen from these 2 parents. Okay, so let me just clear all that. Next slide here is another practice question, but you're going to do this one by yourself. So green peas are dominant while yellow peas are recessive. A yellow pea-filled, I didn't want to say peed plant because I felt that sounded weird. A yellow pea-filled plant is crossed with a homozygous dominant plant. How many of the offspring will be yellow? All right, so I'm going to give you 2 minutes right now. Don't write your answer in the chat yet, even if you think you already know it, right? Most important part of genetics is that you show your work, because you will be surprised how often you think you know the answer and then you do a Punnett square and you go, oh, I was wrong. Or sometimes you find out you were right. So set up a Punnett square here. Which alleles will the parents have? What will the offspring be? How many will be yellow? Go ahead and start doing that by yourself. Okay, if you have your answer already, you interpreted the vocab, you Punnett squared it out. I see a couple people suggesting, Elizabeth or Lane says 1, so you mean 25%, Lane? That's what I'm guessing, right? Remember, we express this as a percentage because they're not having 4 kids. We want to know what percent of their kids. They may have 100 kids, in which case 25 of them would be this, right? So you always want it to be a percent so you could apply it to any number. We got multiple people saying 25%. Elizabeth says none. Any other thoughts here? Marissa also thinks 25%. Okay, so let's break it down. I'm not going to tell you guys what the answer is right away. What would our parents be? What about our yellow parent? Little t's. In this case, though, we're looking at green and yellow, so we want to use a letter appropriate to that. So we're going to use G's because green is dominant, so big G will be dominant. So 2 little g's will be our yellow plant. And then what about our homozygous dominant plant? What would the alleles be for that plant? Big G's is our parent 2, right? Or homozygous dominant. Okay, so now if I do a Punnett square of these guys, I'm going to have big G, and then on the side, I'm going to have my little g's. It looks more like a Y, but you'll have to go with my ugly mouse handwriting. And guess what? I get 100% big G, little g, which I'm not going to write in all the boxes, but you would see, you would have 0 yellow plants. They would all be green because all of them would have 1 big G. So that is a pattern you will notice. That's what happens when you have homozygous dominant, homozygous recessive, cross them together. Guess what? 100% of the offspring will be the same. For example, let's say that you have a parent who has a widow's peak and they have 2 dominant alleles, and your other parent doesn't have a widow's peak. All the siblings will have a widow's peak because every single one of them will be getting a dominant allele from one parent. Right, so that happens. Now, what if we crossed 2 of the offspring? So now what would happen if we crossed G, big G, little g times big G, little g? How many of our second generation of plants would be yellow? And again, don't be afraid to write it out, right? I know you think in your mind, oh, I could just do this quickly in my head. Sure you can. Write it out though, just to double check. Okay, Lane says, I have a question. Would it be if they had 100 kids, would it be 25 yellow, or would it be random for each plant and each child would have a 25% chance of being yellow? That's a good question, Lane. Technically, it would be that they have a 25% chance of being that. But in reality, if you looked at a large number of people, you should see those numbers. So it would be both, technically. However, right, in genetic counseling, which is a career where you do this type of work with people trying to find out if their child would have disease, right, you would say, for example, they have a 50% chance of having the disease, right? And if that's true, then a lot of people wouldn't want to have a kid, maybe, because that's a pretty high chance to some. All right, Marissa says 75% are yellow. What are we agreeing with that? How many yellow plants would we have if we crossed two heterozygous plants? 25%. So 75% would be green. 25% would be yellow. Oh yeah, you thought green. Well, that's why you said 75, right? So again, in that case, you have 75% green. However, they're not all the same, right? We have one homozygous dominant, two heterozygous, and one homozygous recessive. So again, this is the kind of stuff Mendel did, and he just wrote these things down. Because let me tell you, if you're an amateur gardener, right, and you have a yellow pea plant and a green pea plant, you would think that if you cross them together, you're going to get some yellow, right? That's what I would think. But that doesn't happen. Pretty interesting stuff. Maybe not. Maybe you're like, this is boring. All right, well, just in case you're bored, it's going to get a little harder now. So that was the easy ones. Clear all drawing. Okay, now we're going to talk about if we have more than one trait. So another thing that Mendel found out, we'll come back to this one. Another thing that was found out by Mendel is that traits are inherited independently. Hold on, I'm gonna have to answer this phone, which I'm gonna do while I take you guys over there so that, oh, nope, it gave up. Okay, never mind. So Mendel also found out that both traits of an organism, two traits of an organism, right, can be inherited independently. In other words, dominant traits don't go together and, you know what I mean? Like, they don't separate in certain ways. It's totally randomized from meiosis, right? Something that we've already studied and talked about. So in this case, we're going to try to figure out what would happen if we have two traits, right? Which is much more realistic, because usually are we just wanting to predict one thing? No, well, we want a dog or a plant that has 18 things that we want, right? So how do we get that? Well, here's a great example of that. Dogs can be black, dominant, big B, or brown, little b, as well as spotted, giant S, or smooth, tiny s. A brown smooth dog is mated with a heterozygous mate. What percent of their offspring would be black and spotted, smooth and black, and brown and smooth? Okay. Notice my Punnett square just grew a lot in size. Now, I'm going to show you how not to do this. This is what people do when they have not seen how to do this. And you're going to tell me what's wrong with it. Oh, come on, text box. So, hello, Mr. Ling. We're doing a dihybrid cross right now.
Sweet.
This is what people do that's wrong. Tell me why it's wrong. Or you can say, I have no idea, but I'm glad I know that it's wrong because I will never do it. Because it's going across. Okay, well, yeah, but they would also have another one. So let's just pretend. This is not following exactly what the question would say, by the way.
Wait, dihybrid two traits? Or dihybrid?
Dihybrid two traits.
Two traits.
Unrelated.
Okay, fine.
Okay, so most of you are probably thinking, that looks good. I would do that. No! Erase it from your mind. It is wrong. Why? Because you're telling me that out of two traits, this dog isn't getting one of them. This dog would just be a color and not have fur. How is that going to work? Where's the spots or the smooth? We're talking about two traits here. So you're going to tell me that dog is only getting one of the traits, but the other dog is getting the other trait. No, right? So yes, we need to figure out how can we set this up. Remember what we're working with here. We're working with alleles. So first, let's figure out what our dogs actually are. So if we have a brown, smooth dog, what does that mean about their letters? Well, brown is little b's, and smooth is little s's. So we have a completely homozygous recessive dog here, brown and smooth, being crossed with a heterozygous mate, meaning all traits are heterozygous, so they're going to have a big B, big S, little s. Now, alleles separate independently, but they do separate. So that means from dog parent number 1, the possible alleles that I could get are, I could get the first little b and the first little s. I could get the first little b and the second little s. You guys like that I picked BS? You could get the second little b and the first little s, or you could get the second little b and the second little s, which in this case, it's the same, right? So you might think, it won't be though for the second dog. So that means our first parent, we would write BS, little b, little s, for all of their potential alleles. Now, they're actually giving an allele for each trait. Because remember, you don't want to just give one gene and just completely ignore the other one. Okay, what about our second dog? What potential gametes could our second dog produce? Well, we could have a big B and a big S. We could have a big B and a little s. We could have a little b and a big S. Or we could have a little b and a little s. Right? So what am I doing? Literally the way I do it is I go first, 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 last. Right? It's almost like foiling. Second, first, second, last. Right? It's almost like a FOIL situation in algebra, except it's way easier because it doesn't actually involve math. Just thinking in the same way. So those are the potential alleles that I could have for my second parent. So now I would write those on the top. Again, the top or bottom, honestly, it doesn't matter, right? Just keep your parents straight because you don't want to end up mixing up the alleles because then you'll get the wrong answer. Also, I usually over-exaggerate my size of my S's just because it can be confusing to figure out if you drew a big S or a little s. Good advice for the future. Okay, so then now what would I do? I would just fill in all the boxes, which I'm going to do with typing because that'll be easier. So I'll have a big B, little b here, and a big S, little s here. This box, I'll have a big B, little b, big S, little s. Guess what? I'm going to have the same thing. Maybe I can copy paste. Nope. Doesn't let me do that because it's Zoom and it sucks, but oh well. Big B, little b, big S, little s. Big B, little b, big S, little s. Okay. Second column. Big B, little s, little s. And same thing all the way down. Third column. Little b, little s, little s. Again, it's going to be the same thing all the way down. Oh, that's a big S. Just kidding. Erase that. See, I'm telling you, S's are hard. They're the hardest letter, I think. So this one will be little b, big S, little s, all the way down. And our final choice here is little b, little s, all the way down. Now, it doesn't always work out where there's so many boxes in common. Like, for example, we only have 4 options here, right? That just happened to work out because of the cross that I set up. If you really want to practice this one, do 2 heterozygous parents. You can even do this dog example, but do 2 heterozygous parents and you will see it could be a bunch of stuff all over the place. However, we're out of 16 boxes now, so our percentages will be slightly different. In this case, right? We wanted to know what percent of the offspring would be black and spotted. Well, to be black and spotted, we need a big B and a big S. So where do we have a big B and a big S? Only in this first column, right? So 25% of our dogs would be black and spotted. How about smooth and black? How many of our dogs would be smooth and black? What do you guys think? 50%? Well, let's see. To be smooth and black, which is the first one that I asked. Oh, I said black and spotted the first time. Whoops. You're right. It's 25%. I kind of screwed that up. I did the wrong one first. So, excuse me. Over here is our black spotted dogs. And over here is our black smooth dogs. Okay. Gets confusing. How many of our dogs would be brown and smooth? 25%. I know, the S's are hard. I'm telling you, they are tough. Probably should have picked a better letter to represent this, but you guys see the point of this. Right, so in this case, we just have, 25, 25, 25, 25, right? It's nice and in fourths. Again, if you really want to challenge yourself to make sure that you've understood this concept, try a heterozygous cross. That is the most complicated one. And again, it gets hard because there's a lot of letters, there's a lot to look at. You have to think about what you're interpreting. Okay, let's go back to the widow's peak. And, okay, bye Elijah, thank you. Let's try this one. A widow's peak is dominant, right? We already said that. John is claiming that he must be adopted because he doesn't have a widow's peak, but both of his parents do. Is he right? How do you know? Also, super intense thing to do to your parents. I think I'm adopted because I analyzed my genetic traits and it doesn't seem to add up. Stressful times. So what do you guys think? A widow's peak is dominant. So where should we start? Let's start with John. What must John's alleles be if he doesn't have a widow's peak? Little W's, right? He has 2 recessive alleles. That's how he's going to get no widow's peak. Great. Well, his parents both have widow's peaks, so they could be big W's, right? Or they could be, as Lane suggested, heterozygous, big W, little w, right? They could be either or. Now, for them to produce a child like John who doesn't have a widow's peak, which ones must they have been? You could do a reverse Punnett square here, right? Or you could look at the alleles and go, well, there's no way that a parent with 2 dominant alleles gave a recessive allele, right? So you are right. His parents must have been big W times big W, little w. And then there's a 25% chance that they would have produced a child with no widow's peak, which would be John. So John is not right. He's wrong. Now, if he did a detailed genetic analysis of his parents and he found out that one of them was big W, okay, well, that is a little concerning, right? But there's really no way to know. It's without doing a genetic test, there's no way for you to know if you are heterozygous or homozygous dominant. It's almost impossible to tell. Sometimes in certain traits, they'll be more pronounced, right? So like sometimes some people have like a really extreme widow's peak. Usually it is thought that they must have 2 dominant alleles. Not always true, right? Same with the PTC tasting. If you have a heterozygous genotype, supposedly it's like a little bit less bad than if you have 2 dominant ones, right? Again, I don't know. I can't taste it, so I don't even know what it tastes like in the first place. But you can assume that, right? But there's really no way to tell in that case because the dominant trait's being expressed. So you don't really know what the second trait is. But in this case, you know that it's possible that they could have made John. All right. Final opportunity to do a dihybrid cross with some deer. Oh yeah, cilantro tasting. So similar concept, right? It's PTC is linked to bitter tastes. So if you hate cilantro, right, that's a very specific bitter flavor. Potentially could be genetic. I don't know 100% if that is genetic. There's some things that aren't. Actually, you know what? I'm going to give you guys this question for extra credit. So if you'd like extra credit in the class, or you just want to do it because it's fun, because it is, I'm going to post this PowerPoint, do this question. In fact, I'll change the slide to say extra credit in giant letters. If you turn in an answer to the question and your work, and you're right, then I will give you 15 bonus points on anything of your choice. It's pretty nice, right? Let me repeat that in case you weren't paying attention. 15 bonus points for doing this question, submitting it to me. Just email it to me. It's fine. With your answer and your work, most importantly, right? I want to see your dihybrid cross. You should have 16 squares. You should have alleles. Remember, there's 2 traits. They need to be inheriting both. Okay. So that's an extra credit thing you can do. There is an assignment linked to this that you should be able to do now. In fact, it asks you about your widow's peak genotype. So that's in Canvas already. The PowerPoint's also in Canvas. I forgot, I already posted this. Woo, I'm on top of it. So I'm going to change this to extra credit. And I'm going to make that only people who attended today. All right. And because for the rest of the virtual session, as much as I want to do another practice problem with you guys, I want to talk about what's coming up for the rest of the semester, which I know is important to a lot of you guys. First question, when does the semester end? Excellent guess from Colin, 31st. Yep, that's when it ends. Right, so that's a lot of time. Today is the 8th, yes. So you have almost a month. So if you're struggling to catch up or other things, right, you have time to fix that and get your grade where you'd like it. Otherwise, I finally published a whole module ahead of time. I'm so proud. The entire module is published, everything that you're going to need to do. I think I will add, oh yeah, I'm working on grading, Marissa. Everything's there, right, that you need to do. Another thing that you may want, which again, you can email me about, if you want me to put zeros in for you, I can zero out the whole class for you so that you can see what your grade is live, right? That's really easy for me to do. If you would like that, just send me an email. That way, if you're trying to get to a certain grade, you can see exactly, maybe it's getting to a passing grade, maybe it's getting to a C, whatever. So that's an option for you too. Otherwise, I won't do that to you, right? Because I know some people are like, oh, you put zeros in and I haven't done it. Anyway, a lot of this genetic stuff is thinking. It's thoughtful. Is it right to do this? Would you do this? Is it okay for this to happen? Our next virtual session next week is actually going to be our last one. We will do pedigrees, which are another way to look at traits being passed down. Again, you might have done them before, super interesting. We will talk a little bit about X-linked diseases. However, we're not going to go into too much detail on that. There are some pretty heavy morality questions in this class for you. And then most importantly, the thing that I really want to talk about is your final project for this module, which is a genetic disorder project. Should be an interesting one. And it's worth 100 points because it's a pretty short module. So it's worth a lot of points. There are a lot of diseases or disorders that you can pick from, right? They're all here and they're all linked. So I'll paste this in the chat right now so that you can look at it right now if you'd like. But I encourage you to go and pick a disease that maybe you don't know a lot about or that you think you kind of know about it, but you're not sure. Like, for example, Cri du Chat. I'm like, what could that be? That sounds like a weird disease, right? I'm going to click on it. And I'm going to see what it is. It's also known as 5P syndrome and Capgras syndrome. It is a rare genetic condition that is caused by the deletion, a missing piece of genetic material on the small arm of chromosome 5. The cause of this rare chromosomal deletion is unknown. What are the symptoms? Right? The symptoms are that they have a high-pitched cat-like cry. This is real. Mental retardation, delayed development, distinctive facial features, small head size, widely spaced eyes, low birth weight, weak muscles. The cat-like cry typically becomes less apparent with time, right? Most of them have difficulty with language, blah, 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 right? So, for example, you probably never heard of this disease or disorder. Maybe you have heard of it, right? But maybe that's what you want to do your project on. But this is the kind of stuff that we're going to be breaking down. What does that mean to have a deletion on the P arm of chromosome 5? You'll see some of that stuff in the next lesson. Now, I'm telling you about the project now because you really don't need much more background to be able to complete the project. So there's no reason that you can't start picking what you would like to do. The only one that I removed from their website list is autism, just because that's a pretty new one to be added to the list. Most scientists agree that it's definitely genetic related, whether it's environmentally caused that then changes the genes or if it's inherited, right, remains to be seen. So that one doesn't have a lot of information, so I took that one off the list. Again, if you have something that's not on the list, like Bonnie, I know you're probably going to want to do your own genetics, right? That's possible, but I want you to branch out a little bit, especially if it's something that you just wrote a paper about. Pick a different one. There's the requirements of the project. It could be any form you want. It could be an essay, a PowerPoint, a video, a brochure, something else. Whatever form you would like is totally fine. But yeah, so keep in mind that's going to be the last thing that you have to do for the class, and it is going to be do the final thing. I believe I made it January 25th. Is that what I did? Load. There we go. Nope, January 28th. Okay, so January 28th, your project is due. Otherwise, the rest of these assignments are pretty short. They shouldn't take you a lot of time. There's no more tests or quizzes, right? It's just writing, basically, for the rest of this. All right, does anybody have any questions? Oh, I'm also really excited about this biotechnology assignment. Oh, okay, well, there you go. That's right, you wrote that in your paper. I'm sorry, Bonnie. I'm having a difficult time getting back into the swing of school after my lovely relaxing break, as you can tell. Well, good, so you're gonna research something else, and I'm gonna be very interested to see what you pick. So this is another assignment that I'm pretty hyped about. Mostly I'm just really excited about all these assignments that I made for you guys. But you're gonna be researching biotechnology, including something called designer babies. Right, so you're gonna pick one of those things to research. So that's pretty interesting. So it's a lot of research, self-interest driven stuff here in this last module. Great way to end out the semester. All right, so if you don't have any questions, right, you have the assignment that goes with today's virtual session. And we're going to have one more session next week. And then that's pretty much it. I will leave you to your own devices for the end of the semester, and then second semester we'll resume our virtual sessions again. All right, so thank you guys for being here today, and I will see you next week. Oh, interesting, Lane. Yep, that's the thing that happens. All right, bye everyone.